Welcome back, class. So in chapter 8, we're talking about genetics. We're going to talk about DNA replication. We're going to talk about transcription and translation. Hopefully those are terms that are somewhat familiar to you. And if they are not, then I'm going to suggest that you pause this video and go back. There is an introduction to genetics video, especially for you to kind of help you get a feel for what we're talking about. I want you to start with that discussion first. But if you're comfortable with transcription, translation, and replication, then let's jump in. We're also going to talk about mutations. We're going to talk about horizontal gene flow. That's something pretty unique to bacteria, so you may not have talked about that much before. So, some vocabulary words. I'm going to let you pause and read these terms, so I'm not sitting here just reading it off the screen to you. Okay, here's some more terms. Pause the video and read them to you. Make sure especially you can tell the difference between genotype and phenotype. And make sure you could describe what's the difference between a plasmid and the bacterial chromosome. The chromosome being the big main holder with the genes that are required for bacteria to live. Plasmids being small little things that can replicate on their own, so you could have lots of little plasmids in one cell. And typically, plasmids are things that are not required for the cell to survive. Okay, pause, make sure you have these terminology straight, and then meet me on the next slide. Okay, the genetics is so central to understanding our cells that this concept is called the Central Dogma. The Central Dogma tells that DNA, which cells always have, is used to create messenger RNA. Messenger RNA is used to create protein. And proteins in your cell are the little worker bees who give the function to your cell. So you have DNA to be able to make freckles. That leads to RNA, which leads to the protein, and the protein actually leads to having freckles on your skin. Or you have DNA to be able to digest sugar, which leads to RNA, which leads to the proteins that are actually digesting sugar. But what if your DNA is mutated? Maybe someone has been exposed to sunlight and it's mutated their DNA. Maybe just chance, evolution just is chancing to mutate something. A mutated DNA means you're going to have mutated messenger RNA, which means you're going to have a mutated protein and an altered function. It could be that mutation makes you lose a function that's necessary. It could be harmful. It could be that mutation leads to a new function that could give a species a new ability to survive, a new advantage. It could be that altered function is just different. Instead of having brown hair, you have blonde hair. Not necessarily advantageous or harmful, but just different. There's different types of mutations we're going to talk about, mainly base substitutions and frame shift mutations. So by the end of this chapter, you'll be able to tell me all about this screen. So here is your little parent cell, your little parent bacteria. In DNA replication is copied. So this one chromosome is copied into two. So you have two chromosomes to make two daughter cells. So each daughter cell has its own DNA. In gene expression, this is going to involve your DNA being used to make RNA, being used to make proteins. That's gene expression. That gives you the protein so the cell can metabolize. It can break down energy sources. It can build up things that it needs. It can grow. This process, here's two new words for us. The process of using DNA to make RNA is called transcription. The process of RNA being used to make protein is called translation. So I think of transcription makes me think of a court transcriptionist. So a court transcriptionist is there in court typing down what people say. They're speaking in English. She's typing in English. It's the same language, just different versions of it. It's the same thing with DNA and RNA. They're both nucleotides. It's the same language, just different versions of it. DNA is kind of a more stable, sits around your cell forever, used to, hold the information in the nucleus or in the nucleoid, where RNA is a more short-term region, goes out to be kind of the middleman. But translation would be changing to a whole new language. So in translation, we're switching from the language of RNA, of nucleotides, to proteins, to amino acid language. There's also recombination. Do you remember touching on recombination in chapter 1? I hope you do, because we're going to talk about it again. Recombination is when two different cells swap out or combine their genetic information. So here you can see this cell has this nice dark purple DNA, but in this recombinant cell you have dark purple with a light purple. Bacterial cells can just swap out information on their own. They can do that. We also can do it intentionally in a lab. We might stick in a gene for a protein that we want to make. So maybe you're trying to synthesize cellulose or something for some sort of mechanical, for some sort of economic reason. Or maybe you are trying to grow some or produce some insulin or something for a medical treatment. You can stick in a gene for insulin. This recombinant cell then will start expressing that gene that you just combined in there, and you'll have that new product. So here's this key concept. DNA is a blueprint for a cell's proteins. That's what we saw here. It is a blueprint. It is what controls how RNA is made, which controls what proteins are made. So it is a blueprint for proteins. It's obtained from the parent cell. It can be obtained, the parent cell then swaps it down to its daughter cells. It can be obtained from another cell if we're talking about recombination. DNA is a double-stranded molecule. So in this picture, we have two strands, this strand and this strand. They will twist together in a double helix. And those two strands, their bases are going to be joined together by hydrogen bonds. So we have A and T here, joined together by a hydrogen bond. G and C held together by a hydrogen bond. In DNA, whenever you have an A on one strand, you always have a T on the other strand. So if there was a T here, there would be an A here. Whenever you have a G, it's always bonded to a C. So here there's a G here and a C here. There's a G here and a C here. So if you have the sequence for one strand, you can know exactly what the other sequence is going to be on the other strand, because you know all of your A's are bonded to T's, all of your T's are bonded to A's, all of your G's are bonded to C's, and all of your C's are bonded to G's. That's an important concept. And there will be a time when I'll ask you, here's one strand of DNA, what would be the strand that it's bonded to? If you're looking at this, they have sugars on them, and their sugars have 5 carbons. So as this cartoon lights up, you can watch it say 1, 2, 3, 4, 5. There are 5 carbons. And on this 5' carbon, so this little dash means this 5' carbon is where the phosphate group is, this little green circle. And these two strands run in opposite directions. So here on this strand, you can see the oxygens. These O's are facing up. This is running 5' to 3'. That means that the 5' carbon is at the top, and this is the 3' carbon here at the bottom. This other strand, the oxygens are facing down. It's running the other direction. So here, this is the 3' carbon at the top, and this is the 5' carbon at the bottom. That's called anti-parallel. I think of the two strands as two lines of students lined up. So imagine we're in class and we just do something like this. If you're facing opposite ways, both lines are still made of people. They're still the exact same thing. They're holding hands to represent these hydrogen bonds. But if they were to let go and start walking, they're going to walk in opposite directions. So even though they are identical in composition, because they are facing opposite directions, they're going to move in opposite directions. They're going to act a little bit differently. And when we see DNA, when we see it open up and start getting copied, we're going to see those two strands are going to copy in opposite directions because those enzymes are going to move in opposite directions along DNA. Okay, at this point, you can tell me, why is base pairing in DNA important? You can describe to me what bases will pair with which bases in DNA. You can give me a clinical application of genomics. So especially with that, we mainly discussed recombinant DNA, how that can be clinically applied. You can describe to me the anti-parallel nature of DNA. That's going to be important also. Okay, I will see you back in our next chapter. We're going to, or our next video. We're going to start talking about how DNA copies itself. So every time you have new daughter cells, how are they getting DNA? Okay, see you later. Bye.